Welcome to Nucleus second part, second video. Here we are going to talk about the structure of nucleus. Structure of nucleus. Structurally, nucleus has different parts, four parts mainly. Four parts structurally. What are the four parts? Let us look at this. First part is nuclear envelope. Second is nucleoplasm. Third is chromatin. Fourth is nucleus. So structurally, nucleus has been divided into four parts. What are those? First one is a nuclear envelope. Second one is nucleoplasm. Third one is chromatin. Fourth one is nucleus. So when talking the first one that is nuclear envelope. Nuclear envelope. As I told you in the beginning, nucleus is a double membranous cell organelle. So if it is a nucleus, look at this. This is the nucleus. It is a double membranous cell organelle. Outer membrane and inner membrane. So this is outer membrane. This is outer membrane. Outer membrane. This is inner membrane. Inner membrane. Outer membrane is called ectokaryotheca. Outer membrane is called ectokaryotheca. Outer membrane is called ectokaryotheca. Inner membrane is called endokaryotheca. Endokaryotheca. And space between the two membranes is called peri-nuclear space. Peri-nuclear space. So the space is measured about 10 to 50 nanometers. 10 to 50 nanometers in size. The space is measured about 10 to 50 nanometers. 10 to 50 nanometers. And each nuclear membrane, the thickness of each nuclear membrane is about 5 to 10 nanometers in thickness. 5 to 10 nanometers in thickness. Means the size of this nuclear envelope, means the thickness of this nuclear envelope is about 5 to 10 nanometers in thickness. 5 to 10 nanometers in thickness. And yes, outer membrane will be continue with endoplasmic reticulum. Outer membrane will be continuing with the endoplasmic reticulum. Suppose this outer membrane, it is continuing with the part of our endoplasmic reticulum. This is endoplasmic reticulum. Yeah. Outer membrane is continuing with the endoplasmic reticulum. Endoplasmic reticulum. Outer membrane is continuing with the endoplasmic reticulum. Okay. And as this membranes, both the membranes, they are made up of with the smooth membranes. Outer membrane and inner membrane of nucleus is made up of with smooth membranes. Smooth membranes. Outer membrane and inner membrane both are made up of with smooth membranes. As they are moving parallelly each other, there are many what you can say perforations will be seen in this membrane. Many perforations will be seen. Because of these perforations, outer membrane and inner membrane will be overlapped. Outer membrane and inner membrane at certain places they will be overlapped. As they overlap, there is an opening spot will be formed. This opening spot will be called nuclear pore. Nuclear pore. So outer membrane and inner, due to these perforations, outer membrane and inner membrane will be overlap and form a opening space will be called nuclear pore. So the number of nuclear pores are about 100 to 10 power 7 in numbers. 100 to 10 power 7 in numbers, and the diameter of this nuclear pore is about 10 to 100 nanometers in diameter. The nuclear pore 100 to 10 power 7 in number, and the diameter of nuclear pore is about 10 to 100 nanometers in diameter. Next, in this nuclear pore, there are cartwheel-like diaphragm will be plugged off. In this each nuclear pore, there are cartwheel-like diaphragm will be plugged off. This cartwheel-like, this is a cartwheel. So this is the structure. It is plugged at each nuclear pore. It is a cartwheel-like structure. This is, what is this? Cartwheel-like structure, which is plugged at each nuclear pore. This cartwheel-like structure will be called annulus. Cartwheel-like structure will be called annulus. So this annulus will be act as a plug for this nuclear pores. So because of this nuclear plug, what is happening? Whenever this annulus is open, then the content which is present in the nuclear membrane will be coming and contact with the outer cytoplasm. Whatever the content is present in the cytoplasm will be exchanged into the nuclear plasm through this nuclear pores. So that means these nuclear pores will help for exchanging of nuclear matter with the nuclear material with the cytoplasm, and materials from cytoplasm into nuclear plasm. So, and that is the main function of this nuclear envelope. So when talking about nuclear envelope, it is a double membranous cell organelle. Outer membrane is called ectokaryotheca. Inner membrane is called endokaryotheca. Okay, outer membrane is called ectokaryotheca. Inner is called endokaryotheca. And between these two membranes, there is a space is present. Is called Peri-Nuclear Space, which is about 10 to 15 nanometers in diameter. Diameter means length, space. And then this outer nuclear membrane means ectokaryotheca will be continued with the endoplasmic reticulum in the cytoplasm of the cell. And where ribosomes will be get attached. And then these outer membrane and inner membrane will be made by smooth membranes. As they move in parallel to each other, there are a number of perforations could be formed in these membranes. That means the outer membrane and the outer nuclear membrane and the inner nuclear membrane will be overlapping. Because of these overlapping, there are many perforations. Because of this perforation, there is a space is provided. Those space will be called nuclear pores. The diameter of this nuclear pore is about 10 to 100 in nanometers. 10 to 100 in nanometers. The number of this nuclear pores about 100 to 10 power 7 in number. And at each nuclear pore, there is a cartwheel-like diaphragm will be present. And this structure is known as an annulus. Annulus will be act as a plug for this nuclear pores. Whenever this plug open, the nuclear material is exchanged with the cytoplasm and vice versa. That means cytoplasm to nuclear plasm through this nuclear pores. Yes, that is about the nuclear envelope. Yes, understood? Yes, next nucleoplasm. Nucleoplasm, it is transparent granular homogeneous fluid or matrix of nucleus. Inside this nuclear membrane, there is transparent granular homogeneous fluid or matrix is formed, will be called a nuclear plasm. This material is called nuclear plasm. Nucleoplasm. This nucleoplasm, also called karyoplasm. This is slightly acidic in nature. This karyoplasm is otherwise called nuclear sac. Or karyolymph. Nuclear sac or karyolymph. This is acidic in nature, right? This fluid which is present here, it is slightly acidic in nature, and this nuclear plasm contain the materials like DNA, RNA, proteins, DNA, RNA, proteins, and as well as carbohydrates, lipids, and apart from all this, what are present? The enzymes like DNA polymerase enzyme, and DNA polymerase enzyme, RNA polymerase enzyme, DNA polymerase enzyme, and RNA polymerase enzyme, and dipeptidases, and NAD synthetase, etc. are present in this. And along with these materials, what are the materials are present in the matrix of nucleus? The materials like DNA, RNA, proteins, carbohydrates, lipids, and glycoproteins also. We can write glycoproteins, and ribonucleoproteins, also present in the matrix of this nucleus. Apart from this, there are enzymes like DNA polymerase enzyme, RNA polymerase enzyme, and DNA synthetase, peptidases, etc. These are all present in the matrix of nucleus. What is matrix of nucleus? Look at this. This is nuclear plasm. It is a transparent, granular, homogeneous fluid or matrix of the nucleus, which is called a nuclear plasm, otherwise called a karyoplasm. It is slightly acidic in nature, also called nuclear sac or caroline. This is contain DNA, RNA, proteins, carbohydrates, lipids, glycoproteins, and ribonuclear proteins also present. Along with this, the enzymes like DNA, RNA, polymerase enzymes, NAD synthetase, and dipeptidases, phosphatases, sulfatases also will be present in this. Also will be present in the matrix of this one. So this is about the nuclear plasm of the cell, nuclear plasm of the nucleus. Yes, this is about that. And next part. Next part, that is this one, chromatin. Chromatin is highly coiled, thread-like, darkly stained material which is present in the nuclear plasm. Is called chromatin. This chromatin, it is formed as a thread-like structures in this. Formed as a thread-like material in this. This is called a chromatin. This chromatin will form as, normally it will be formed as a reticular structure like this, coiled reticular structure. So therefore it is called a chromatin reticulum. During interphase, the chromatin reticulum will be absorbed in the nucleus. The chromatin reticulum will be absorbed in this nucleus, in the nuclear plasm of the nucleus, during which stage? Interphase. So, interphase is considered as a stable phase where the nuclear material, which is called chromatin, will be seen. As I told you about this, the term chromatin was given by Walter Fleming. The term chromatin was given by Walter Fleming. The term chromatin was given by Walter Fleming. So, what is chromatin? It is highly coiled thread-like, darkly stained reticular structures which are present in the nuclear plasm of the nucleus will be called a chromatin. This chromatin could be absorbed stably in interface, during the interface of solution. And the term chromatin was given by Walter Fleming. Suppose in case of eukaryotic, in case of eukaryote, in case of eukaryotic chromatin, this chromatin is composed by RNA, chromatin is composed by DNA, proteins, and trace amounts of RNA are present as the components of chromatin. DNA, proteins, and trace amounts of RNA will be present in this chromatin material. So that is the composition of chromatin material in this one. Okay, and here in this eukaryotic chromatin, the proteins, there is a specialized proteins which are called histone proteins are composed. Histone proteins are composed with this eukaryotic chromatin material. But whereas in prokaryotic chromatin, these histone proteins are lack of histone proteins. So devoid of histone proteins are present. Lack of histone proteins are present. That's why it is called prokaryotic chromatin. In prokaryotic chromatin, histone proteins are absent. Histone proteins are not present in this prokaryotic chromatin. But in the eukaryotic chromatin, what are present? Histone proteins are present. Basically, histone proteins are made by basic amino acids. There is a rich amounts of basic amino acids are present, such as lysine and arginine. And the basic amino acids. So the rich amounts of lysine and arginine are present in the chromatin material of eukaryotic cells. Those proteins are called histone proteins. Suppose if these proteins, there are histone proteins are about 5 in number. What are those? H1, H2A, H2B, H3 and H4 histone proteins are present. There are 5 histone proteins are present. Now these histone proteins will be seen in nucleosome structure. What is nucleosome structure? In this, there are 4 histone proteins are present as a core, and the under core there are 4 histone proteins are present. In these histone proteins, there is a H2A, H2B, and H3, H4 will be present as a core, and here also H2A, H2B, H3, H4 are present. So these core histone proteins are wrapped by DNA threads, DNA material. So there is a 200 base pair length of DNA is wrapped around each histone proteins twice. Between these 2 core histone proteins, there is a spacer DNA is present. This is called spacer DNA is present, or linker DNA, spacer DNA or linker DNA. This linker DNA will be called integral DNA. What is this? Integral DNA. So this integral DNA is bound by H1 histone protein. Okay. So about 200 base pairs about 200 base pairs. 54 base pairs will be yes, 54 base pairs will be 54 base pairs will be the spacer DNA, and remaining wrapped DNA threads around this core histone proteins is about 146 base pairs. 146 base pairs. Okay. So this is about the nucleosome model. The term nucleosome model was coined by Auret et al. It was discovered by Kornberg. It was discovered by Kornberg and J.W. Thomas. This means this nucleosome model was proposed by Kornberg and J.W. Thomas, and the term nucleosome was coined by Auret et al. So this nucleosome model was a universally accepted model about the description of histone proteins and DNA threads. DNA is a negative charge molecule. Histone proteins are positive charge molecule. So there is a meaningful bounding will be there between histone proteins and the DNA material. So that is the reason why this nucleosome model was universally accepted model about to describe how the DNA and histone proteins are beaded in a nucleus. Yes. So that is about the histone proteins. These histone proteins are present in the eukaryotic chromatin, but not in the prokaryotic chromatin. In prokaryotic chromatin, these are absent. But apart from this, there are other histone proteins also present. What are those other histone proteins? So look at this. Means these are called non-histone proteins. Non-histone proteins. These non-histone proteins are made by neutral amino acids and acidic amino acids. And the number of these histone proteins are non-histone proteins are about the range of non-histone proteins in the cells is about 12 to 120 in number. 12 to 120 in number. Usually, these are present as a constant in the enzymes. What are those enzymes? DNA polymerase enzyme. So, this DNA polymerase enzyme is composed by non-histone proteins and as well as RNA polymerase enzyme. About this, out of these two, the RNA polymerase enzyme is a major enzyme which is present in the eukaryotic cells, eukaryotic and prokaryotic cells. So that is it is a non-histone proteinated enzyme. Abundant non-histone proteinated enzyme is RNA polymerase enzyme. RNA polymerase enzyme. Now, so when you conduct Robert Fuelgen was prepared a staining dye. Robert Fuelgen was prepared a staining dye which is called acetocarmine or Fuelgen reaction. According to this staining material, in the DNA, in this chromatin material, we can see two distinctive regions. One distinctive region is called euchromatin. The other one will be called heterochromatin. Euchromatin, heterochromatin. According to this acetocarmine dye or Fuelgen reaction. So, the differential staining parts could be seen in the chromatin. That's why it is called heteropyknosis. heteropignosis. This differential staining is called heteropignosis. In this, we can see euchromatin and heterochromatin. Euchromatin is slightly stained fibrillar part of chromatin material. It is lightly stained fibrillar part of chromatin material, and it is loosely coiled chromatin material and contain active genes. Such parts of the chromatin is called euchromatin. Heterochromatin is a darkly stained part, and it is a granular part, and this contains inactive genes. The term heterochromatin was given by Emil Each. The term heterochromatin was given by Emil Each. So, by Robert Feldgen was given a staining material which is called acetocarbon and Feldgen reaction. Because of this reaction, then we can see that two distinctive regions in the chromatin will be called heteropignosis, and one is called euchromatin, the other one will be called heterochromatin. Euchromatin is lightly stained part, fibrillar part, and loosely coiled larger part, bulk part of chromatin, and it contains active genes. But this one it is a darkly stained granular part, and it contains inactive. The term heterochromatin is given by Emil Each. So this is about chromatin material. So the next we can see that Nucleus next video. All the best.